Hey folks. On today's video, we're going to be talking about our different types of technologies that have been created for different types of genetic technology. So the first one we're going to talk about is DNA fingerprinting. So essentially what happens here is crime scene investigators would retrieve samples of tissues, could be hair, all these different DNA samples from a crime scene. And so what they would do at this point is they would mix them up and they would extract the DNA from a solution. And so they extract these long strands of DNA and they add an enzyme, and that enzyme is going to cut up the DNA into smaller fragments. And now those fragments are then going to be combined and added to a gel, and it's going to create a pattern. So eventually what happens is then they're transferred to photographic paper, and then you can look at, say, potential suspects, you can look at victim, you can look at evidence from the crime scene, all those things, and you can try to make matches. So if you take a look at this image here, you'll actually recognize suspect 2, and it looks like evidence from the crime scene. These two are connected, and they look like they're a match. So pretty amazing what we can do with regards to these technologies, but that's DNA fingerprinting, so you'll want to know that process. Now, the next one that we're going to talk about is transgenics, and transgenics is going to be your GMOs, so your genetically modified organisms. So a great example of this is frost-resistant tomatoes. So you add the gene for frost resistance into the tomato, you get frost-resistant tomatoes. So it's pretty neat what they can do. There's negative ways that we can see this being used, though. So one example would be bioweapons. So genetically modified organisms designed to infect people and make them sick or kill them. Okay, so obviously not a good way, but again, that is still GMOs, and that is still transgenics. So it's transferring genes from one species to another, and that's essentially what it is. So we'll move on to gene splicing. And in gene splicing, this to me always kind of blows me away when I see this. The fact that this is happening is you're taking bacteria. So for example, insulin producing bacteria in the pancreas. So what's happening in gene splicing or recombinant DNA is that you're taking a plasmid. So a plasmid is out the nucleus of a bacteria. And so you take this little plasmid and you cut a section out of it. And so when you cut that section out, you can see that plasmid, cut that section out. Now you take a section of this gene responsible for producing insulin, and now you actually add it to that plasmid. And so now what happens is that plasmid's now placed back in that DNA, and it just starts to produce and replicate and produce mass amounts of insulin, because it's producing the amounts of insulin that a person couldn't create or produce before. So that's gene splicing. So plasmid is cut, gene is produced that produces insulin, is then placed out into that plasmid. It's then placed back into a bacteria, and then that bacteria begins to produce tons of insulin. It's an amazing process. And another one, there's two more we'll talk about, but the next one's called gene therapy. So again, these show up on your quizzes, your unit exams, but they also show up on the diploma exam as well. So you'll want to make sure that you understand these and the difference. So gene therapy, basically what happens here is you take a vector like a virus, for example, and you're going to use that to essentially to cure or replace a defective gene. So you're going to take this healthy gene right here. So say for cystic fibrosis, you're going to take this healthy gene and you're going to inject it into a virus. Okay, so now it's going to go inside this virus here. So now what happens is then you place the virus inside this person, and eventually what happens is it is going to start treating, that virus is going to start treating this cystic fibrosis. Okay, so now that healthy gene starts going in the lungs, and it starts replacing those bad cells as well, and eventually it's going to cure the lungs as well. But we inject this into a virus, inject the virus into the human, and then it starts working and producing healthy cells that essentially are going to cure us. So looking at the next section, what we need to talk about is bacteria and how they become resistant to things like antibiotics. So basically, if we don't take our whole prescription, then what happens is there's leftover bacteria, and so now they have the opportunity to mutate, and if they mutate, those survivors mutate, then they build up resistance to that particular antibiotic. Okay, so it's important that if the doctor says take your antibiotics, 2 pills for 7 days, they give you 14 pills, you take 14 pills, because if you don't, there's a chance that you're allowing bacteria to mutate, and next time your antibiotic is not effective. Okay, so the last thing we are going to talk about is, again, just the form of, it's called transformation. So it's just another form of bacterial resistance that we'll see. And sorry guys. And anyways, what happens here is it's just you're transferring fragments from one DNA. So bacteria is transferring DNA over to another living bacteria. So it might be a dead bacteria, but it's transferring to a living bacteria. So now what's happening is if you have anything, if it's got any mutations and such, or any resistance in that dead bacteria, then it's passing that along. And now greater diversity within that bacteria is going to create a greater chance for resistance. So again, those are different types of technologies, but also talk a little bit about bacteria and how they become resistant to antibiotics and such. So anyways, hopefully the video is helpful, and we will see you in chemistry next.